Thank you, Brandy. It's a pleasure to follow both Alicia and Bill onto the stage. And what I'd like to do is really pose some bigger questions that address the work that we do about basic science and more in terms of the clinical work. And that's what I was asked to do, is this is a panel discussion following this. I've got my funding listed here. That's the only disclosures I have. But for an outline of my talk, these are sort of the 4 questions that I think they've been touched on in both of the presentations we've had before. How much to reduce salt intake in a human population? What's the evidence for that? What are the problems and the holes in the data we currently have in the literature? And Bill showed this very elegantly. It's not easy to assess the salt sensitivity of blood pressure in a human. What are the flaws in the processes that we have with that? What are possible future ways to address this and make it easier for clinicians and to do diagnoses? Alicia touched on this briefly, and I think it's a very important point. Future direction of clinical trials. We've heard about the GenSalt trial. We've heard about DASH sodium. Doing those in today's academic environment is costly and probably prohibitive. And then finally, I think this is very important in the context of guidelines. Every single one of us in here knows we should eat less salt. None of us do it. We're honest, right? So how can we get this message across? And we have a leading expert here who can input on this, and we can talk about it in the panel discussion. Alicia was mentioning ratios of sodium to potassium. I'm not going to look at a candy bar and figure out the ratio. Neither are you. How can we get this out to a lay public level? You may, but I wouldn't. So the first question I have here, and this is a really important one, how much should we be trying to reduce salt intake in our diet? We all know it's bad for us. We've all seen the evidence. And this is just a statement I've picked up, lifted from the AHA scientific statement in 2016. The salt sensitivity of blood pressure carries prognostic implications, potentially as those as strong as traditional cardiovascular risk factors. So it's a huge public health impact. We've heard about multiple studies, and this study has been referenced before by the other speakers. It's basically the association of urinary sodium and potassium excretion with blood pressure. Major study published in the New England Journal of Medicine. And as you can see here, the average excretion was about 5 grams of salt per day. That's way in excess of the current recommendations, as I've illustrated for you here. In red, you have the American Heart Association guidelines recommend about 1.5 grams a day. The World Health Organization recommend about 2. And as we've heard, the new DRI guidelines fall just above this here at about 2.3 grams per day. So this is you and I. This is America, I think. This is the global average. So there is a great discrepancy between these numbers we all know and what is actually happening in reality. And in terms of dietary sodium restriction, the bigger question becomes, where are we going? This is a beautiful study showing, again, this is the same study showing the direct association with increased excretion of sodium in the urine driving up blood pressure. It's a very well-known, very well-established relationship. I don't think there's any real controversy there. The controversy comes in the next couple of slides, which have been presented by both Dr. Madonna and Farquhar. The J-shaped curve. How much salt in our diet is safe? What are the recommended reductions? So I won't labor on these. This is one version of the slide published back in 2011 in Diabetes Care. This is another version of the slide published in JAMA in 2011. And again, you can see this is our sweet spot. This is us right now. The current recommendations, you could argue, are taking you off into this not safe part where there's an increased cardiovascular risk. This is controversial, and there is still argument in the literature. So this was a study published by very famous people, some of them may be in the room, in Circulation in 2004, getting away from this J-shaped curve, showing a nice linear relationship. And the key that I want to draw your attention to is just down here. This was taken from the TOHP study, and they're talking about the methodological challenges of prior studies, including PURE and other ones, combined with the overall health benefits. They recommend you can reduce sodium intake to the recommended levels in the diet, the recommended guideline levels. And then there was a study in 2018, again, based on the PURE cohort that has caused some controversy. Even there were some publications on it this week that I'll talk about in a minute, where their overall summary, I just want to draw your attention to here, is they say sodium intake was associated with cardiovascular disease and stroke only in communities where the mean intake of salt was greater than 5 grams per day. They're recommending a different strategy. A strategy of sodium reduction in communities and countries where the sodium intake is above 5 grams per day might be appropriate. So they're trying to get into the stratification. The bigger question becomes in this, and it was alluded to by Bill and by Alicia. How do you assess sodium intake in a human population on a large scale? So the PURE study has been criticized heavily, and I will refer you to this reference here. This is where I've basically got these 3 points. These aren't my arguments. I'm presenting the current controversies in the field. This is a very nice counterpoint discussion, I would say, going on in The Lancet that was published earlier last week, arguing about the flaws in the current methodology used. Spot urine in the Kawasaki formula has some issues. How can you take a single spot urine sample and assess sodium excretion? Do you want to take total 24-hour urine? Do you need to do multiple 24-hour urine samples? The DRI guidelines recommend 2 24-hour urine samples to assess sodium excretion, and from that extrapolate intake. It is suggested in these counterpoint articles you actually need 7 days of consecutive 24-hour urinary excretion to get a real estimate of what's going on in a patient. Clinically, that is not very realistic. I don't think any of us are going to walk around with a bucket collecting our pee for a week. And there is this other concept that's been put forward in there. The suggestion of something called early morning fasting urine may be more accurate because you will collect everything that was generated overnight, and that is thought to be, by some groups, a more accurate representation of the sodium intake. I don't have any answers. I'm putting these questions out here for panel discussion and from the feedback from the audience. So it strikes me that as a field, we don't actually agree on how to even measure how much sodium is being intaken in our diet globally. So that's a methodological problem that needs to be addressed to move this field forward. This is another question I think is important. How do we assess the salt sensitivity of blood pressure? The current gold standard would be a 21-day dietary sodium intake where we wash you out and measure your blood pressure. Bill talked about this too. This is the classical 29-day protocol for determining salt sensitivity, where you have a normal sodium diet for 14 days, and then you switch through the low, high, normal salt diets and look for a change in blood pressure. This is very laborious clinically. An abbreviated protocol has been proposed for a 5-day protocol where you have acclimatization, and I've included the parameters for you here. This is still a 5-day protocol. This is not a standard way your clinician, I can't take Brandy and lock her up for 5 days and measure the salt sensitivity of her blood pressure. She wouldn't like that. So clinically, as a field, it would be better if we had a more rapid and reliable way to address the salt sensitivity of blood pressure. And this has been noted, and there are guidelines listed in the American Heart Scientific Statements put out by the NHLBI as well. It's well acknowledged that the diagnosis of salt sensitivity is cumbersome, poorly standardized, and rarely performed in the clinical setting. That's a bit of a problem. A rapid and reliable biomarker for the salt sensitivity of blood pressure is kind of a holy grail. I do a little bit of work on it. I'm sure many people in this room do. It would enable us to do these targeted recommendations. We could do cardiovascular risk stratification, do mechanistic studies the same way that Bill is doing in his defined salt-resistant versus salt-sensitive populations. It'd be great if you could do a screening that would take you a day and identify a patient population. It would speed up the time it takes you to do your research. And then we could then assess drug efficacy in salt-resistant versus salt-sensitive patients. One of the concepts that I like is the tying of genetic data to mechanistic pathways. So I'm just going to show you 2 studies that have done that briefly. One is from the lab of Dr. Matson, and this was published in Hypertension, and it's too tiny for me to see the year on here. I think this was back in 2014. They took the results of a GWAS screening study looking at a mutation in SH2B3 from a genome-wide association study. What they did is they then looked at mutations in this gene in the dull salt-sensitive phenotype. So they went from a big GWAS study down to a mechanistic study in a well-known established model of salt sensitivity. What they found is by doing this mutation, you impacted the salt sensitivity of the blood pressure. By combining human genetic approaches with a basic science approach, they were able to provide novel genetic insight into an inflammatory mechanism in both hypertension and renal-focused disease, showing the power of a human genetic program being combined with a basic science research program. My laboratory did it the other way. I work on GI2 proteins in the brain. They're a G-protein coupled receptor signal transduction protein. We've published for many years, if you take these out of the brains of a normotensive salt-resistant animal, you make the animal become salt-resistant. They have an important role. We went back and we found mutations in this GNI2 gene have been linked with hypertension in 2 cohorts. We wanted to look at the salt sensitivity of blood pressure. Thankfully, the GenSalt dataset is available online through the dbGaP resource. I don't know if many of you are aware of that. You can screen the genetic data. This is a very boring slide with lots of numbers, but really what I wanted to show you is we were able to find a single nucleotide polymorphism in this gene that we've done mechanistic studies on in animals correlated with the salt sensitivity of blood pressure in the GenSalt cohort. So we've gone from a basic science standpoint to human genetic studies to try and show that GNI2 polymorphic variance is a potential biomarker of the salt sensitivity of blood pressure in a specific subset of patients. This is restricted to the GenSalt Chinese Han cohort. We are doing more studies now to broaden this to other genetic backgrounds, and hopefully we'll have results for that soon. I would just like to talk about one other profile that I thought was pretty exciting. This came from the lab of Binna Zhou. And some of you may have seen this. It was published in Cell Metabolism, I think. I can't read it here. Very recently. And what she did is their lab took dol salt-sensitive animals on a low or a high-salt diet and did metabolic profiling. And you can see very clearly here, there is a very different metabolic profile when these animals are challenged with a low or a high-salt intake. And you can see there are different metabolites being increased in response to a high-salt diet. This could be a very interesting way to broaden the way we are looking for biomarkers. And in this paper, I urge you to read it. They took it from this generalized discrepancy to a very specific metabolite that they followed through in mechanistic pathways, again showing the power of screening down to a very specific molecular pathway. So the future of biomarkers, I don't know where this is going. Urinary biomarkers could be very powerful. I'm sure Dr. Madonna knows there's there have been several studies looking at urinary exosomes, so the markers of renal sodium transporters such as the NCC. The Ellison lab have done some powerful work in this. So exosomes, urinary biomarkers, the utility of microRNAs may come into play. And then I've included for you here pharmacogenomics and metabolomics, or lipidomics. Could be very important in building up a profile to distinguish a salt-sensitive versus a salt-resistant phenotype. The next point ties to clinical trials. So I've shown for you here, what is the next generation clinical trial going to be? We've had these very, very big studies, the GenSalt study, the Dietary Approaches to Stop Hypertension study. Doing these in the current climate would be pretty much prohibitive. I don't think you'd get a study section to give you the money to do it. So how are we going to move this forward? I know Dr. Madonna is a proponent of the smaller clinical trials, such as the size that Dr. Farquhar is doing. These may be not powered to the same extent as PURE with 60, 70,000 patients. We don't all have that accessibility. There is nothing wrong with smaller trials that provide mechanistic answers. And again, this is a very controversial topic, and this was published in Hypertension recently, the call for clinical trials, asking or targeting a way to address dietary sodium intake, as Dr. Madonna and Bill both illustrated. It needs to be done in a very controlled, very structured way. And this sort of committee came up with some recommendations that potentially this could be done in prison populations. I'm not a proponent for or against this. I'm presenting the viewpoints that were covered in this editorial because it's an intriguing question and this is supposed to be a controversial panel discussion. The advantages that were stated by this group, I can tell by the reactions in the room, this is a very controversial subject, which is why I'm presenting it here. The advantages that were given for trying to conduct a sodium intake study in a prison population. You have the ability to control a large part of the dietary intake. Dr. Farquhar will tell you how hard it is to do a dietary intake study to ensure that your patients are eating what they need to be eating. You have a large, diverse, multi-site population in the US prison system. That's true. Prison systems do, and this was a surprise to me, they have an existing infrastructure, including IRB boards. And you have the opportunity to do a large randomized trial design. Obviously, the negatives, we're all aware of this. The ethics of prison research is very, very sensitive. And the ability to recruit prison sites is questionable. So the final part that I want to talk to before we get to the panel discussion, and I think this is really important, is how do we reduce population-level salt impact or intake? All of us knows we should eat less salt. We don't do it. We're not very good at branding this. And so this is something I found that I thought was a very neat way of doing this. This is from the World Health Organization, and it's titled Shake the Salt Habit. And it touches on several of the subjects we've seen. I'll skip over that. So this shake package from the World Health Organization envisages a world where the average salt intake, and I thought this number was interesting given the controversial paper that just came out based on the PURE study, is they want to reduce salt intake to less than 5 grams per day for adults and children. They say if this is achieved, it will result in major health benefits and reduce non-communicable disease burden. But what they do is they give a very simple way that ties into everything that's been talked about here today. They talk about surveillance. So you measure and monitor salt use and salt intake. They're trying to bring industry in. Promote the reformulation of foods and meals to contain less salt and potentially, for Dr. Madonna, more potassium, bringing those sort of things in. Adopt standards, and I think this is important for labeling and marketing. Increase knowledge at all levels, not just in a room of scientists like this, at the community level, to your grandmother, to your kids if you have them. And also the environment. And I will quickly go through this because I think it's important. They talk about specific interventions in here, measuring and monitoring population salt consumption patterns, because we still don't have a clear handle on this. Measure and monitor sodium content in food, and evaluate any impact of a potential salt reduction program. There is a link through this with industry. Set target levels for the amount of salt in foods. There is actually a charter or a political declaration by the United Nations on this that I've included for you here. And in terms of the labeling, this was touched on by Dr. Madonna. I'm never going to pay any attention to this. It's cumbersome. I don't read it. I'm sure you don't. But I might pay attention to something like this, a big red salt shaker or with a slash through it. Keeping the message simple and clear so anybody can understand it without having to take the time to look at it. Even these variations are better, where this is showing you this is bad, it's in red, and look at the amount of salt that's in there. A clear color-coded system or something like that is suggested in this document. There's a big component on education, integrated education and communication about health risks and dietary salt and dietary sources of salt at all levels, high schools, workplaces, things like that. And then, this is the last one, will be kind of an intervention, an environmental intervention to make this possible. And this is where it gets to the community outreach, public health workers, nurse practitioners, doing this in schools, workplaces, hospitals, taking it out of the academic environment in a room in Orlando, Florida. Because then the general public aren't listening to us right now. With that, I'm going to finish, and I think we get to move on to the panel discussion. Thank you for your attention. I appreciate it.